First of all, thank you to the organizers for inviting me. I'm very honored to speak here about extracellular vesicles. I hope I can convince you that these are very novel and exciting new biomarkers and bioactivators, and I'm convinced that you will hear from them later. So I will give first an introduction and talk a little bit about EV biogenesis and the detection for those in the room who are not very familiar with this topic, and then talk about EVs in AKI injury and repair. Actually, most of the data is in AKI repair, and the data was also mostly generated here in Italy. Let me share some cryo-electron microscopy images from our own urine. You see here a really very heterogeneous, oh, I'm sorry, I knew this would happen. You see here a very heterogeneous group of vesicles. These extracellular vesicles are submicron, less than 1 micron. We have various sizes from very small, less than 100, they're also called exosomes, to larger ones from 100 to 1,000 nanometers, which are called microvesicles, microparticles. We have, they're mostly circular, but we have also different shapes, different densities. Here you also see some uromodulin in the urine. You find these vesicles in all kinds of body fluids. They are, for the kidneys doctors, they are in the urine, they're in the blood, they're in peritoneal fluid. We even find them in the interstitium from kidney. The interest in these vesicles is really exponentially growing, especially during the last 2 decades. And we even have now an International Society of Extracellular Vesicles. The last meeting was this May in Barcelona. They had over 1,000 attendees. I already used 3 names for these vesicles, so the nomenclature is really confusing. Most of the researchers use the word exosomes, but more and more we are using the word, it's more a generic term, extracellular vesicles. Because we enrich for one group or the other, but in the end we have a mixed group. So I think it's better to use a generic term, and I will do that during the talk. Let me talk a little bit about history. So going back, maybe already in the 40s and 60s, people were describing EVs. Peter Wolf actually described them as platelet dust, and likely described the larger vesicles, the microparticles. And then in the 80s, 2 groups, Harding and Rose Johnson, they were working in parallel on the biogenesis of exosomes, and they published within 2 weeks. And especially Harding and Stahl were interested in studying the transferrin receptor and the maturation of reticulocytes. And what they did is they labeled the transferrin receptor with gold, and they discovered the biogenesis or release of exosomes. They found that these transferrin receptors were within little vesicles and excreted from these multivesicular bodies. Later in the 90s, Clotilde Thierry and Grassa Raposo described the molecular characterization of dendritic cell-derived EVs, so in the immune world. And they had actually the first exosome meeting in 2011 in Paris, and afterwards the International Society was founded. EV biogenesis is really complex, and I could talk 1 or 2 hours about that, but there is really a nice, very recent review paper in Nature Reviews by Grassa Raposo and Guillaume Van Niel, which summarizes this. But for today, let me just simplify the biogenesis. We think there are at least 2 ways these vesicles are generated. The larger ones, between 100 and 1,000 nanometers, are generated through a blabbing process. There is a rearrangement of the phospholipids, and calcium and enzymes are involved, and these vesicles are shed from the cells. And they carry markers of the parent cells, that's how we identify them, but they also carry cargo, proteins, lipids, nucleic acid. In contrast to the microvesicles, the larger vesicles, we have the exosomes, which are released from multivesicular bodies inside the cells. They also carry markers of the parent cells and have cargo. And in contrast to those 2, there are some apoptotic bodies, of which we know much longer. They are generated during the programmed cell death. These vesicles are released by each cell in health and disease. So depending on the stimulus, different vesicles are released. And the composition is really, really diverse. It depends what kind of stimulus we have. And this busy slide is just summarizing how diverse it is and how many different types of proteins these vesicles carry. And there is really no clear EV marker. And there are some markers you find more in exosomes, they are on the larger ones, but I think it's justified to use still a generic term. They interact with recipient cells that makes them so unique. They're not just biomarkers, they have a functional role, and there are different ways how they're internalized and into the other cells through phagocytosis, lipid wraps, or membrane fusion. And there's more and more data showing their functional role in different fields, in immune modulation, hemostasis, angiogenesis, matrix modulation, proliferation, adhesion, tissue generation, so also in therapy. You can imagine that because they are so diverse and heterogeneous, that the detection is quite tricky, and they are also very small. There are now recommendations by the international societies that you need to be really, you need to provide a comprehensive analysis with a couple of techniques to describe these vesicles well, because you want to know what you put in your Petri dish. You want to understand what they're doing. You need to know what you put in your Petri dish. So you need to describe different categories of proteins, EV and non-proteins. So you detect the EV mimics. You have to size them, count them. You have to image them. And they have also specific suggestions for functional studies, especially with emphasis on controls. And the larger vesicles are mostly studied with flow cytometry, but there are other techniques using Brownian motion to detect size and the Coulter principle to also detect size and concentration. In our lab, we have now established a rigorous analysis. I'm doing this now for 10 years, and it has been really a journey. We size and count these vesicles. We provide good images. We also start to do targeted phenotyping. So I'm interested in EVs deriving from kidney cells, so I can pinpoint and define the origin coming from the glomerulum or tubulus, for example. We do Western blot analysis, but we also do mass spectrometry and qPCR to analyze the genetic content. As a kidney doctor, I think the urine is an ideal body fluid to study these vesicles. They derive from all cell linings in the urinary tract and carry markers of the parent cell, from the glomerulus to all the tubular cells. They cross the glomerular filtration barrier, so they can be reflective of systemic effects. And we learn more and more they participate in intranephron communication. So when you have glomerular damage, why do you have interstitial fibrosis? And there is more data coming. And I have one example. Dylan Berger from Ottawa, he is interested why when in diabetes you have the glomerular damage, you also have the interstitial fibrosis. So what he did is it's a very simple experiment. He took podocyte-derived EVs and added them to human proximal tubular cells and measured markers of fibrosis, fibronectin, collagen 4, and then when he added those vesicles, you see an increase, but when he added TGF-beta inhibitor, there was a decrease. So now let's talk about extracellular vesicles and kidney injury. These EVs have a diagnostic role. They can have a prognostic role, functional role, but also a therapeutic role. That's why they are so unique and fascinating. The first studies have focused on the prognostic role. And very early on, actually in 2001, people started to characterize these vesicles in the circulating. So from where are they coming in the blood cells? In patients with severe multi-organ failure, including with kidney injury during sepsis, they had, for example, lower platelet-derived EVs and tissue factor-positive EVs, but higher granulocyte and erythrocyte-derived EVs. So this is kind of descriptive. Another study a little later showed that monocyte and plate-derived EVs are elevating AKI in sepsis. So you think, oh, this is a little contradictory. Yes, I think the verdict is not out. It's descriptive, but it's also due to the fact that these were smaller studies. The techniques were different, and people were not using very sophisticated tools. And these groups studying EVs in the circulation focused mostly on the larger vesicles. People have also looked in urine, and a lot of researchers in AKI are still on the hunt for the ideal biomarkers. So you're wondering, are finally these EV delivering our acute kidney injury biomarker? So one group early on as well checked NHE exchanger type 3 in urine exosome pellets. So they focused more on the smaller ones, interestingly, in the urine. And they found them were high in ATN, but not in pre-renal azotemia in 68 patients admitted to the ICU. So this is kind of a diagnostic role of these vesicles. In a rat model of cisplatin-induced AKI, only the exosomal phytion alpha, which is a filtered protein metabolized in the proximal tubular cells, but not the free phytin, was detectable in urine before kidney injury. So the way you generate these EVs is you spin down the urine, you have your pellet, and you have the supernatant. So it was in the pellet, but not in the supernatant. And a similar result was found when they tested NGAL in urinary exosomes in patients with delayed graft function after transplant. They couldn't find a urinary free-floating NGAL. So this might indicate that potential biomarkers for AKI might be enriched in these vesicles, and we don't find them free-floating in their soluble form. This is one of the latest studies. The other ones were earlier, from last year, where the researchers looked in a larger group. And what they did is they wanted first find out if this marker is of interest. So they had an animal model. They induced AKI and sepsis with cecal ligation and then ruptured later this, and found out that activating transcriptional factor 3 is early on activated in ATN. So they thought, okay, let's translate that to a human model. And they studied this in a prospective way and in a multi-center study that had over 100 patients actually. And they compared urinary NGAL in the kind of supernatant versus the exosomal fraction for transcriptional factor 3. And they found that this is kind of a timeline here. When the creatinine rises, the red line, this is NGAL, this is AFT3, that NGAL was nicely early on elevated, came down, but AFT3 was even higher and came down fast. And the ROC curve performance was better for AFT. I think the verdict is still out. This is descriptive, but I think it's encouraging. So let's talk about EVs in therapy. So they seem to be ideal. EVs are already natural mediators to numerous biological processes. They're already there. They likely have a lower immunogenicity than cells. They're stable. We could have a high delivery capacity, and there would be also less risk with cell graft and engraftment and proliferation. There's even more. EVs could provide a very targeted therapy because specific EVs could bind to specific cells. This is already accomplished and moved much further in cancer therapy, finding a specific EV which attacks a cancer cell. This could happen through delivery of a drug, a ligand, by mediating a receptor-induced signal, an altered protein, or an RNA. They are actually, with electroporation, they get silencing RNA into these vesicles and can treat disease. Important is also, I think, that EVs are really small enough to cross the blood-brain barrier and the glomerular filtration barrier, which is important for us. Researchers who started who are working on stem cells for treatment of kidney injury, they also thought, oh, maybe we can use EVs arising from these stem cells. And for stem cells, there are already over 46 clinical trials in AKI and CKD, and they showed, oh, okay, it's not there. Oh, it's back. Okay, they showed improvement or stable disease. And these stem cells are multipotent. What's also interesting, just to keep in mind, they're already kidney resident stem cells. So this is the first big paper about stem cells derived in these AKI coming here from Italy, from the group around Professor Camisi. From Turin. They showed a protective effect of EVs deriving from human bone marrow-derived mesenchymal stem cells in tubular epithelial cell of a mouse model, SCID mice with glycerol-induced AKI causing rhabdo. And they labeled these mesenchymal stem cell-derived EVs with a membrane dye and showed that they were incorporated in the tubular cells. They treated these with RNAs, which abrogated the protective effect, suggesting that maybe there is an RNA-dependent mechanism. These are some pictures from their papers. First of all, they showed that they were dealing with vesicles in their preps. And this is the histology after 5 days and 8 days after during the AKI model. And this is, you see earlier on the tubular damage. When you add the microvesicles, there was a protective effect. You had the same effect adding just the stem cells, and this was known. And when you added the RNAs, you kind of diminished this protective effect. They also could show that we had here a transfer of human mRNAs. There was some horizontal transfer. They looked for human proteins in these tubular cells from mouse and detected 2 human proteins here. They showed later on another proof that the microRNAs are important effectors of the paracrine effect of VEVs. They used the fact that Rocher and driver enzymes are crucial enzymes for regulating microRNA maturation inside the cell. So they generated stem cells which were not having these enzymes, kind of knockouts, and they then injected in the previous model regular mesenchymal stem cell-derived EVs and those from these knockouts. And you could see when they added these knockouts, they did not have a protective effect. This is a BUN, cast, and necrosis. So another proof that there is likely a paracrine effect through microRNAs. After this initial experiment, mesenchymal stem cell-derived EVs were tested in other models of AKIs. It's just working for one model or in others. So they showed it works in a lethal cisplatin model and gentamicin, ischemic reperfusion injury. And then there was the question, how much do you have to give, single or multiple injection? So in this lethal model, in the cisplatin model, a single injection ameliorated the renal function pathology, but only multiple ones in the end decreased mortality and restored normal histology in renal function at 21 days in surviving mice. So that was quite interesting. And then they looked into other stem cells, not just the mesenchymal stem cells. They tested EVs coming from endothelial progenitor cells in a rat model, and they could show that it protected against progression of CKD by inhibiting, for example, capillary rarefaction. So when you have AKI, can you prevent the progression to CKD? They used EVs from Wharton jelly, which was reduced inflammation. And then they used kidney-derived stem cells. And in the end, there was another question, which EVs are doing this? We have this heterogeneous group, are the smaller or the larger ones? And I will show you that in the next slides. This slide summarizes or provides the evidence that likely the smaller ones are causing this, but I think the verdict is still out. So when you prepare these EVs, most of the research do differential centrifugation. When you just spin for 10,000 K, you generate likely larger vesicles. Then you take the supernatant, spin at 100,000 G, and you have the smaller ones, the exosomes. Or you go straight to the 100,000. So they had 3 different fractions, the larger ones, smaller ones, and then a mix of all. And actually, the smaller ones showed more protection than the larger ones. This was also shown in a paper by another group, Erdbrügger in Ottawa. But in other disease processes, like in lung ischemia, the larger ones were effective. So I think the verdict is still out here. Now we are coming to the question, are EVs from kidney stem cells directly able to do this? And EVs from all different types of stem cells from the kidney were generated, and in a nice dose response, they had a pro-angiogenic effect. This is the latest study, which I think is also fascinating. Now, here we're looking which stem cell in the kidney is doing this. And again, the Camusi group looked here at glomerular-derived stem cells and tubular-derived stem cells, and they had fibroblasts as controls. There's a sophisticated way to describe all these stem cells, and then they generated the EVs and injected them into their IRI model. And interestingly, when they injected the glomerular mesenchymal cells, they had protection. And when they did inject EVs, they had the same effect. They did not have such a strong effect when they used the tubular-derived stem cells. They did something else here. They tried to clean up this EV prep. So it's kind of a crude method, I told you, by centrifuging these. They cleaned them up with a floating method, And I think for me that's another proof that we are dealing, or that the EVs are the effectors here and, through a paracrine effect. So I'm now speculating maybe EVs directly from the urine could be protective. And there was a recent paper in JASN showing that recombinant temhorsfol injected could decrease inflammation in acute kidney model. And I showed you earlier in my cryo-EM pictures that there are temhorsfol proteins in our PrEPs and they might interact with EVs. So I think that would be the next step for me. So if I summarize mechanism of EVs in repair, it's through a paracrine effect by a horizontal transfer, likely of mRNA, microRNA, and proteins reprogramming their phenotype. And there are different signaling pathways involved affecting fibrosis, angiogenesis. EV therapy, I think, depends on the source of the cell, the type of the population, which injection route, timing, dosing. So there are a lot of unanswered questions. And also challenges. You know, we still don't know if it's safe. If maybe there is an engraftment, and something bad can happen. But overall, I'm very enthusiastic about EVs. They are novel biomarkers and bioactivators that makes them so unique. They are messengers. I think we have to learn more about mechanism in kidney disease, but interestingly within the kidney, but also cross-organ. What happens with all these talks between the kidney and the liver and the heart? These EVs might be messengers there. And there is a therapeutic potential for these EVs in kidney disease. I think it's realistic. It has already been used in chronic kidney disease in a study in Egypt. They injected it through the renal artery and slowed down progression. It's only one study I found. A lot of work needs to be done, especially dissecting the EV role in an AKI mechanism. And I like to thank, again, the organizers and the UVA Department of Medicine, Division of Nephrology at UVA, and my team members. Thank you so much.